To set the stage for everybody, let's start with a brief introduction about FFPE and what it stands for. FFPE is formalin-fixed paraffin-embedded tissue. It's the most common format of archiving solid tissue for diagnostic and surgical pathology. This is done since decades, and it's a well-known procedure for immunohistochemical studies in the past, but more and more it becomes an invaluable resource for oncologists in drug discovery or biomarker identification, because FFPE tissue is simply the biggest source of biological material. So this material allows researchers to correlate a disease state, for example, with the genotype. But when you are working with FFPE tissue, there are some pros and cons you have to consider. Of course, on the pro side, you benefit from vast FFPE tissue archives. They're easy accessible tissue source, so you can order them from biobanks, for example, or from a close hospital. It's a very convenient sample handling and storage because you can work at room temperature. If you compare this to fresh tissue, where you have to work and ship on ice, for example, and within 1 or 2 days. This is not the case when it comes to FFPE tissue. And there are many commercial available kits on the market for the molecular analysis. On the other hand, what you have to consider when you're working with FFPE tissue, the formalin, which is used to fix the cells, introduce crosslinks between nucleic acids and proteins. The fixation leads to DNA fragmentation and RNA fragmentation, as well as to protein denaturation. Plus, there's no global standard for the FFPE tissue preparation. The current workflow when it comes to molecular downstream analysis from FFPE samples is that you classically prepare an FFPE section from a block and you extract the DNA or whatever from this material before you do your molecular analysis. Solid tumors are highly vascularized and infiltrated by stromal cells such as leukocytes, endothelial cells, and fibroblasts. So the amount and composition of those non-tumor cells depends on various factors, including tumor entity and stage or treatment history. So when you want to analyze carcinoma or tumor cells or tumor and you want to really look at the tumor signal, you have the problem that non-tumor cells bias your tumor signal in molecular downstream analysis. There are some manual ways how to enrich tumor cell fraction. One, for example, is the manual macrodissection, where you manually scrape off a tumor area of interest that is circled by a pathologist from a stained slide. The problem here is that you still have infiltrating stromal and immune cells in the tumor area, and by the manual scraping method, you also scratch off normal human cells at the border area. Another method is laser capture microdissection. Here also you work with stained slides under the microscope, and with the help of a laser you can isolate single cells or cell subsets, but these machines are very expensive. You need expensive equipment and supplies, a very well-trained staff, and it's a low throughput system that is very time consuming. What if you could simply digest your FFPE material and isolate your cells of interest? Miltenyi, with its gentleMACS technology, has a way to generate single cells from solid tissue. So from fresh tissue with the gentleMACS dissociators, it is possible to get a single cell suspension with preserved epitopes to do cell isolation or cell analysis downstream. I'd like to give you now a short introduction into this technology before we move forward. With a gentleMACS octodissociator with heaters, you have a fully automated possibility to dissociate your tissue of interest. It has 8 independent positions for high throughput applications, and you have specific programs for the most frequently used tissues. We combine mechanical disruption and enzymatic digestion on the instrument to be most gentle to the cells. So the enzymes soften the tissue, they degrade the extracellular matrix so that we can work with very mild mechanical shearing, and the mechanical shearing disrupts the tissue into small tissue pieces so that we can work with very mild enzymes. By this we are gentle to the cells and we can preserve the epitopes. Mechanical shearing is done by the gentleMACS tubes, which bear a rotor-stator system for the mechanical disruption, and for the enzymatic digestion part we have tissue-specific dissociation kits like the tumor dissociation kit, lung dissociation kit, or brain dissociation kits. And since beginning of the year, we have launched a new tissue dissociation kit, the so-called FFPE Tissue Dissociation Kit. This is fully automated dissociation of up to two 50 micrometer FFPE sections into single cell suspension, and it is validated to preserve the cytoskeletal markers cytokeratin and vimentin. Cytokeratin and vimentin can be used to analyze carcinoma samples because carcinoma develops from epithelial cells. So carcinoma tumor cells contain the epithelial cytokeratin marker, whereas stromal cells and lymphocytes contain the vimentin marker. So you can distinguish in flow the carcinoma cells and the non-tumor cells by using these two markers. The kit has been validated on various carcinoma types, including breast, prostate, colon, lung, or pancreatic carcinoma. The FFPE Tissue Dissociation Kit helps to standardize the complete process from deparaffinization to dissociation because the kit includes a standardized protocol for the deparaffinization and rehydration with reduced incubation times. Typically, you incubate the tissue section for multiple times 5 minutes in different ethanol and xylene concentrations. We could reduce this to just 30 seconds. After the dewaxing, there is a heat-induced antigen retrieval step, and to do that, the kit includes a heat-induced antigen retrieval buffer to reverse formalin-induced modifications of the extracellular matrix so that it is accessible for the dissociation kit or for the dissociation enzymes. And last but not least, the tissue dissociation reagents, of course, are also included in the kit to provide reproducible results in a fully automated fashion with a high throughput system and by preserving important cytoskeletal markers. So to wrap everything up, the FFPE Tissue Dissociation Kit can be used to standardize FFPE carcinoma tissue dissociation into single cell suspension and to preserve important cytoskeletal marker for the analysis and for sorting. We have prepared validated downstream analysis data I'd like to present you now. For the beginning, we have focused on DNA analysis, and I have some data to share with you today on DNAploidy and genome sequencing. Let's start with DNAploidy. DNAploidy is a prognostic marker that you can use to estimate cancer aggressiveness. To do ploidy analysis, the DNA is stained, for example, with DAPI and fluorometrically quantified. The important thing here is to admixture a reference cell, and typically you use patient-derived fibroblasts or lymphocytes because you have to identify the reference peak of diploidy in the histogram. And taking then the DNA histogram, you evaluate the so-called DNA index or DI by dividing the mean fluorescence intensity of one cancer peak by the mean fluorescence intensity of the reference peak. So let's have a look at this histogram here. In green, we have the reference peak of the admixture reference cells at the intensity of 200. And in orange, we have the histogram from carcinoma cells. And we see here there's one peak that is in line with the diploid peak from the reference cells and another peak at a mean fluorescence intensity of 340, telling us that this cancer is aneuploid, and aneuploid cancers are discussed to be biologically more aggressive. But there's one thing, when you work with the FFPE Tissue Dissociation Kit, there's no need of admixture with reference cells because the reference cells are already within the sample. When you stain the single cells after dissociation, you can stain them with anti-cytokeratin, anti-vimentin antibodies. You can use the vimentin positive non-tumor cells as the reference cells. So let's have a look how it can look like. Here we used as a first sample a prostate carcinoma, and we gated out first the duplets, and from the siglets we gated on DAPI positive cells in this red population, and this red population we split it then based on the cytokeratin and vimentin signal into, in orange, the cytokeratin positive carcinoma cells, and in green the vimentin positive non-tumor cells. And then we gated back these two populations on the DNA signal or on the DAPI signal and plotted it in a DNA histogram. So like this, we see the profile of the carcinoma cells overlaid with the signal of the vimentin non-tumor cells. And we see that both peaks are more or less overlying, telling us that this prostate carcinoma is a diploid carcinoma. Let's have a look at another carcinoma sample. In this case, it was a colon carcinoma. First, we gated out the doublets. Then we gated on DAPI-positive cells. And we split this population again into cytokeratin-positive carcinoma cells in orange and vimentin-positive non-tumor cells in green. The DNA signal of this carcinoma is a little different. We have one peak that is overlaying with the reference peak, which represents the diploid state, but there is another peak roughly at an absorbance intensity of 300, telling us that this colon carcinoma is aneuploid and potentially more aggressive than the prostate carcinoma. Let's have a look now to DNA sequencing from FFPE sections. Here I'd like to present once more the classic molecular workflow. You prep the DNA from a complete FFPE section, you extract it, and then you do molecular analysis. But if you're interested really in the tumor signal, it is very important to enrich the tumor cells. And when you dissociate your FFPE section with the FFPE Tissue Dissociation Kit, you can sort now on a flow sorter, for example, cytokeratin-positive carcinoma cells or vimentin-positive non-tumor cells. And these two purified cell populations you can use to extract nucleic acids from before you move on to molecular analysis. In order to assess the DNA quality after dissociation, we have compared the DNA fragmentation and variant frequencies of DNA one time extracted from a complete tissue section without dissociation and DNA extracted from single cells after dissociation with the FFPE Tissue Dissociation Kit. Let's have a look first on the DNA fragmentation. On the left side, we have the complete tissue section without dissociation. On the right hand, we have the single cells after dissociation. And we see that the mean intensity is identical, telling us that the DNA is not getting more fragmented by the dissociation method. If we look to variant frequency, here we looked at the KRAS gene. KRAS is a typical carcinoma gene that gets mutated at this position. And we again compared here the variant frequency of complete tissue sections without dissociation towards single cells of the same carcinoma sample after dissociation. And we see that both variant frequencies are around 14%, which is telling us also here that the dissociation with the FFPE Tissue Dissociation Kit has no effect on the variant frequency as well. When you want to do DNA sequencing, it is important to get enough amount of nucleic acids, of course. To do next-gen sequencing, you typically need minimum 100 nanogram of DNA. So here's just one example where we have prepared DNA from two 50 micrometer sections. Please keep in mind that this is just a number from this sample, and DNA amount strongly depends, of course, on the tissue size and the wax blocks, the cell number that you can isolate from the cells, and of course the age of the sample, since DNA gets degraded over time, it gets oxidized, for example. In this case, we could isolate from two 50 micrometer sections 200,000 DAPI and cytokeratin positive carcinoma cells, and we could isolate them to a purity of 96%. And from this purified population, it was possible to isolate up to 1.7 microgram of DNA. So this is by far enough to do downstream whole genome sequencing or targeted next-gen sequencing. Okay, so let's have a look now how variant frequencies look like from cytokeratin sorted carcinoma cells. In this case, again, we looked on the KRAS mutation, which is on chromosome 12 at this position. And again, the variant frequency of DNA prepped from a complete section was around 14%. Also, the original fraction of the single cells after dissociation was around 14%, but the variant frequency could be enriched after sorting carcinoma cells based on the cytokeratin marker up to 45%. Here I like to explain a bit more the KRAS gene or the KRAS mutation. KRAS mutation is a heterozygous mutation. So that means that the mutation is only on one of the chromosome 12. So one chromosome 12 is still having the wild type allele, whereas on the other chromosome chromosome of one cell is the mutated variant of the KRAS. In other words, if you find a variant frequency of KRAS of 45%, it means 90% of your cells are harboring this mutation. So if you take now in mind that we have a purified carcinoma population of more than 90%, we found this KRAS mutation in almost every tumor cell. So, as I already said, the characterization of genetic aberrations in paraffin-embedded tumor material is often impaired by contaminating normal cells, and we have shown that with the purification of tumor cells, we can on the one hand reduce false negative results derived from admixture with non-tumor cells, and we could enrich the tumor signal significantly. So we have validated downstream analysis from DNAploidy, our genome sequencing, and I'd like to give you an outlook what we have in the pipeline. We are validating now downstream analysis like single-cell DNA sequencing, DNA methylation, which is important for tumor researchers who are interested in epigenetics, and we like to establish immunomagnetic isolation method of cytokeratin and vimentin positive cells using MACS microbeads. Okay, so as a summary of this webinar, now with the FFPE Tissue Dissociation Kits, it is possible to generate a single-cell suspension from FFPE carcinoma, and we preserve the cytoskeletal markers cytokeratin and vimentin. And by using these two markers, you can isolate cytokeratin-positive carcinoma cells or vimentin-positive non-tumor cells to improve molecular tumor analysis. I like to just mention that the FFPE Tissue Dissociation Kit can be also used for any other FFPE tissue, including spleen, skin, and lymph nodes, for example. Okay, with this, I'd like to close the webinar. Thank you for attending this webinar, and I'm now open to take your questions.